So this topic wraps up a couple things that we've seen already. We've talked a lot about chromosomes, we've talked about inheritance, and we've talked about the behavior of chromosomes in meiosis. And this gets to one of those issues where we look at the crossing over of homologous chromosomes and see how that relates to this distance between genes. And before we get into this, just let me define the words we have on here. Autosomal linkage. So we've seen autosomal, those are chromosomes that are not the sex chromosomes. And this concept of linkage, we saw X linkage or sex linkage. When we talk about linkage, we're talking about basically alleles that are inherited associated with something else. So you can have two genes that are linked together. That means that they're more often inherited together, and then you can talk about this in terms of being physically linked, as in a gene is located on a chromosome, located on the same physical chromosome as another gene. And that'll probably be the easiest way to think of this term. So a little review, one of our last topics was the behavior of chromosomes in mitosis and meiosis. And if you like, you can stop the video and quiz yourself on these. Basically, you can look at any one of these mitosis events and determine, um, sorry, what do I want to say here? One of these metaphase events where the chromosomes are lining up along the midpoint of the cell, and you can determine which of these three processes you're looking at. So this one, we've got a metaphase where the homologous chromosomes, red and blue, are paired up next to each other. And that's something we only observe, that's right, in meiosis 1. So this is a meiosis event. We have the replicated chromosomes finding their homologous buddies and arranging next to them. And we also see the effects of some crossing over, which is going to be one of our topics today. So that's unique to meiosis 1. How about this one? We have replicated chromosomes lining up at the middle of the cell, and they're not associating with their homologous partners. So we have big blue, doesn't really care where big red is. So these are homologous, the big red and the big blue are homologous together, but they're not associating together because during mitosis, which is what we see here, blue is going to separate into two halves and red is going to separate into two halves, and they don't need to find each other to make this process work okay. Okay, so linkage, what we're going to talk about today, breaks one of these rules that we established way back in Mendelian genetics. As we introduce that, I usually try to give you a little bit of a caveat that when we're talking about independent assortment, we're talking about genes located on different chromosomes. So if we go all the way back to Gregor Mendel, when he observed genes that were associating independently, assorting independently, they were not located on the same chromosome. And so it's kind of fortunate for him as he was establishing these rules and the independence of the two separate gene outcomes. He wasn't looking at genes that were linked together. So as we've seen before, A and B have no influence over each other. A is on a different chromosome than B, and so big A can go with big B just as easily as big A could go with little b, as we saw during the process of meiosis. And that influenced our probability estimations of how likely it was to produce one certain genotype for a gamete. So getting big A with big B, if you remember, this is one of the possible arrangements. It's not showing all possibilities on this slide. So big A, in this case, is going to the same side of the cell as big B. We don't, just for simplicity, haven't shown the effects of any crossing over. But maybe just really broadly speaking, if these are both random events, A going to the side of the cell, B going to the side of the cell, then it's just as likely that big A will go with big B as it will be that big A goes with little b. So the chance of getting big A and big B is 50% chance of getting big A, 50% chance of getting big B, and out of then 4 possible outcomes, 2 for A, 2 for B, we're going to get 1/4 of those outcomes being big A with big B. So how would it be that big A and little b end up in the same gamete? Well, all we would have to do in that case is to have the B chromosome, this littler chromosome, just randomly end up arranged in the opposite direction so that the little b's are on top and the big B's are on the bottom, right? So that's just completely random, just shuffling out chromosomes. One of the features that gives genetic variation as a result of meiosis. And yeah, that's how that goes. Okay, so independent assortment, we think we talked through this pretty well. So genes assorting their alleles independently from each other. And the assumption here, the basic assumption, if we want to use the rules of probability of 1/4 chance of a certain gamete genotype, then we need to assume that A and B are in fact independent and not linked together. So kind of contrast to that, let's say we have this other gene D, which is located on the same chromosome as A. Now we've got a situation where big A goes to a side of the cell and it's bringing with it big D. So it's not independent anymore. Big A goes to the top of the cell, big D is going to come with it. If big A went to the bottom of the cell, then big D would come with it, right? So this is not random anymore. And we did see also a chance for big A and little d to end up together. That was the result of crossing over. So if we have a pair of homologous chromosomes, with big A in one location, so the A gene in one location, big A on one, little a on the other, and the D chromosome, or the D gene, what did I say before? D gene located at a different position on the chromosome. In this arrangement, right after DNA synthesis, this is what you would get always. So big A with big D. It's possible, as a result of crossing over, sometimes it doesn't take my keystrokes very well here. I think I'll show the result of crossing over. So we saw the process where these two chromosomes can switch parts of themselves. And the result of that, at least in some circumstances, is that the chromosome that's got the big A ends up getting a copy of little d, and the chromosome that's got little a has a copy of big D. So crossing over, as we saw before, is an equal swap of some part of the chromosome. They're physically exchanging material at some location between the A gene and the D gene. And the question becomes, what are the chances that this is going to happen? So how likely is that to happen? And it's not random. And as we'll see, it's going to depend on the distance between those genes. So whatever two genes we're looking at that might be crossing, having crossover between them, if they're farther apart, this is more likely to happen. So before we get into the distance estimation, a little bit more about some terminology here. So one of the terms that we're going to use when we talk about crossing over is this term recombinant. Or recombination is the result, is the process that gets you recombinants. You might have seen recombinant DNA. This term is used sometimes in other contexts. If you have DNA from one organism put into another organism, people use the term recombinant for that as well. So it's kind of a multi-use term that isn't always specific to this process. In this context, we're talking about recombinant as a result of a process that gets us a different combination of alleles than we saw in the parents. So this is a standard genetic cross. We've got one parent with homozygous big A, big B, and another parent with homozygous little a, little b. And so the gametes that they form, this parent can only make one with little a, and I'm sorry, big A and big B. This parent can only make one with little a and little b. And so these are their gamete combinations. So it's also possible in the heterozygous offspring, when it goes to make gametes, that some of those gametes look like the same gametes that the parents would have made, right? So this heterozygous big A, big B, little b individual can make a gamete that has big A, big B, just like, let's say that's mom, or little a, little b, just like dad. And we've already seen this possibility of getting different combinations than were present in the parent. So this isn't new territory for us, but we're going to introduce these terms, non-recombinant and recombinant, as in basically a gamete combination that was not found in the parents. Another way to look at that is we look at the chromosomes. That's what we're going to be looking at in this exercise. So if we assume that A and B are on the same chromosome as part of this topic, I'll go ahead and put this as a diploid individual. So the parent has some chromosomes. Sorry, the left-hand parent has capital letter chromosomes. The right-hand parent has lowercase chromosomes, and then just as before, these big A, big B, or the little a, little b are these non-recombinant types. And the recombinant types would be big A with little b, or little a with big B. So these are possible as a result of crossing over. They would be not a crossing over event in the parents, but actually a crossover in the heterozygous F1 individual. Let's throw one in there real quick. So this individual would inherit big A, big B from one parent, little a, little b from the other parent, but during the process of meiosis can have crossing over that makes these new recombinations. Okay, so another term you could use is to call these the parental gametes, parental genotypes. So these are the same allele combinations that were found in the parents. So here we have parent chromosome red and parent chromosome blue as they were inherited from the parents. So dad's chromosome, mom's chromosome. And if there's no crossing over, then they look entirely red and entirely blue. If there is crossing over, the result of one crossing over event gets you one of these recombinations. And so that would cross over, in this case, just one of the chromatids. So these two replicated chromosomes, after one crossing over, they still got one arm of the chromosome that's all red and one arm of the chromosome that's all blue, and then one that's traded. So that's the result of just one crossing over event. As we'll see, there's actually a lot of crossing over that happens during meiosis. So probably pretty rare you would only observe one crossover event. Another thing we're going to introduce as we move forward here is a notation to keep track of our genes. So we saw before, if you had a heterozygous individual, right, if we had this individual, if they were separate chromosomes, then this individual over here would have, we just say big M slash little m semicolon big D slash little d. And the difference with that notation, the semicolon tells us that we have different chromosomes. So that's kind of the break point in the notation between two separate chromosomes. And this slash was the split between two homologous chromosomes. So alleles for the D gene would be present on those two homologous chromosomes. Alleles for the M gene would be present on two homologous chromosomes. And this new notation tells us that M and D are located on the same chromosome. So now we have this M comma D, which is not the same as the semicolon. This tells us that big M and big D are on the same chromosome arm, or the same physical chromosome, the same homologue of chromosome. And little m, little d are located on the other of those. So the slash is still telling us that we're splitting two sides of there are two homologous chromosomes, but the comma tells us two alleles for different genes that are located on the same homolog. All right, so I've written just a different way to show what's illustrated in the illustration at the right. You can also use this notation to refer to gametes. So big M comma big D is a single chromosome that exists inside of gamete, a haploid gamete, or little m little d, like that. And of course, as a result of crossing over, we could get different combinations. Another question for this is, which parent would we be able to observe crossing over in? Crossing over would happen in both of these parents, but only in the left parent would we actually be able to observe a difference in the kinds of gametes that are made. The contrast on the right here, this parent, if we did have crossing over, that would just put little d on a different chromosome from little d. So they're the same genetically as far as we can tell. And if you switch this little d with that little d, you're basically trading exact information, the exact same identification information across. All right, so we've seen these non-recombinant, recombinant gametes. Terms can refer to the gametes there. So as far as the results of crossing over and linked genes, basically these, one of the earlier pieces of evidence for knowing that genes were linked was they were upsetting these rules, these Mendelian laws that had been determined a while before. And if you've got linked genes, it messes with that independent assortment assumption. So we do one of these examples here. So we've got a case where we're crossing two kinds of plants with different phenotypes in their pollen and their flower color. So purple flowers, red flowers, long pollen, round pollen. And it says up here, these are homozygous. And it hasn't told us anything about the genotype. So we can use our powers of deduction and figure out what these are. Simple question of, and these they have a complete dominance inheritance type. So which allele is dominant? Well, it's whichever one shows up in the F1 individual. So this is consistent with a P for purple. Let's say big P, and then long pollen also showing up in the F1 individuals. So this is purple, long by red round. And that's, I'm going to do a little exaggerated P here for lowercase p and an exaggerated lowercase l. So those were homozygous. Their offspring, big P, big L, little l, we would expect under complete dominance to be purple and long. And then it says we do self-fertilization. So we're going to take this individual and produce its gametes. So big P with big L, big P with little p with big L, little p with little l. And big P with big L, big P with little p with big L, little p with little l. Let's see if we can just maybe cut to the chase. You could fill this in on your own time if you want. Anybody in these locations? So following the rows and the columns that have all capital P's and L's. Here's one that's got capital P with a capital L. There's a capital P with a capital L there. 1, 2, 3, 4, 5, 6, 7, 8, 9. Did I get that? 4, 5, 6, 7, 8, 9. Yes. So 9 with the P something, L something. Big P only without a big L is these three. So P, L, L. Little p with a big L. Do some triangles there. So 3, little p, big L, and 1 of the little p with little l homozygous. So in our expectations, we would expect 9/16 of these to be purple and long, 3/16 of these to be purple and round, 3/16 to be red and long, 1/16 to be red and round. And this is what we actually would observe in the offspring. And I think maybe rather than running the numbers on this, we could just kind of do a relative expectation. So the most purple and long, that's true. The least would be red and round. Oh no! So this category, which is supposed to have only 1/16 of our outcome, is coming out bigger than this category or this category. And what's going on there is that to get this kind of gamete and this kind of gamete and this kind of gamete and this kind of gamete, all of those require some crossing over. So um, anything that is producing one of these X's or triangle shapes requires a little bit of crossing over. So we got crossing over from that column. And there's a little bit more of these outcomes. So I'm running out of shapes here. This outcome of no crossing over is a big one. This one is no crossing over. That's a big one. This one's no crossing over. So those all contribute to this purple and long. And then this one over here is no crossing over with the all lowercase recessive alleles. That would give us the red round. So there's a little bit of a skew here to outcomes that result from no crossing over, and a lot less of what we would expect in the outcomes that would require crossing over. So basically, if you take the assumption that there's no crossing over, that these alleles are inherited independently, that's not going to hold up in these cases. So you have to kind of figure out what's going on. It turns out that these alleles are getting inherited together for the flower color gene and for the pollen shape gene. Okay, so I think we probably worked through most of this. There's another way you could take this a little more thoroughly and look at the actual numbers of offspring. I don't know if we're ready for that just yet. That gets into something related to what we'll do in lab. Okay, so determining sort of how linked genes are is a matter of, you want to get at that with some number data. And so a good way to get data that can tell you something about your linkage is to do a test cross. So this example here from your book has a couple examples of what happens if there is no crossing over whatsoever. Then these two possible gametes are the only things that could be made by this parent here. So if there's no crossing over, then this heterozygous parent gives you a big M with a big D or little m with a little d. And that's a little bit simpler to work out. It says there's half of the offspring result from this gamete and half the offspring result from this gamete. So that's like extreme linkage where there's nothing in between. There's all homozygous dominant gametes or homozygous recessive gametes, or I guess I shouldn't say homozygous, big M with big D or little m with little d. In this other example, if they're completely unlinked, as we would expect for genes on different chromosomes, then we get that 1/4, 1/4, 1/4, 4 equally likely outcomes. So test cross is a nice way to see what you would expect in the outcome. All right, so here's a case a little more realistic. If you do have linked genes, there's going to be a chance for crossing over to happen between them. So we got some cases of sometimes gametes where no crossing over happened, and other cases where crossing over did happen, as we saw with that one illustration. Some of the crossed over chromosomes, some of the chromosomes involved in crossing over produce recombinant gametes over here. Some produce non-recombinant gametes. What we also see is much different relationship of the numbers of offspring that we'd expect. So we expect if they're unlinked to be 1/4, 1/4, 1/4, 1/4, and what we see instead is a whole bunch of this kind and not so many of this kind. All right, another consideration that's something to think about is that crossing over could happen more than once. So one of these over here, an even number of crossover events between two loci could also get you a genotype for the gamete that doesn't look like a recombinant genotype. Let me spell this out a little bit more. I think I've wandered a little far from what is making sense to you right now. All right, so let's say we've got a couple of chromosomes. Clean slate here. I'm going to lie them on their side so we can illustrate maybe a little bit easier the crossing over possibility. So let's just put our alleles on here. And let's just do A, and I don't know, F. A lot of the illustrations so far have given you the impression maybe that capital letters always have to stick together. Not the case at all. So here we've got one with little a and big F, or big A and little f. So crossing over, as we saw, would happen, would cause these alleles to recombine if crossing over happened at some place in the location between A and F. So one of these crossovers would switch the big F with the little f, and that would give you a chromatid that's got big A with big F together. So that would cause a crossover. But what happens if you had two crossovers? So that would swap them and then swap them back. So big A with big F, but then switch again, big A with little f. So an even number of crossover events in the space between two genes would cause them to look like they didn't actually have a recombination event. And that certainly happens. So that's possible, even number of crossovers. If you're crossing over anywhere on the chromosome that doesn't happen between the genes you're looking at, you wouldn't notice that. So as I mentioned before, in genetics, we're often looking at just a few genes at a time. And so if something happens that doesn't affect the genes we're looking at, we don't see it. We're not prepared to look at that in the outcomes. So one more way that you can have crossing over happen and not have it affect the genotype, as in you'd still have non-recombinant gametes, that would be if you just had the gametes made from parts of the chromosome that weren't involved in the crossing over. So these other chromatids in this example were not involved, and little a is still with big F, big A is still with little f. So the question I put on the slide here, mostly to get you thinking about the process. So if you can determine what would cause recombination to happen between two genes, then you can kind of also think about what would maybe not cause recombination to happen, or cause recombination that isn't visible. So I guess wouldn't look like recombination even if crossing over happened. Okay, so crossing over twice at this in this example, 2 times crossing over would return things back to the way they were. So big M with big D, little m with little d. So those two possibilities, big M, big D, little m, little d, those would both be possible if 2 crossing overs happen. All right, now maybe looking at the concept of the test cross and the gametes coming from each parent here. So we have this one parent, which is big M, big D. I'm going to write this in a little simpler way. If you have the luxury of being able to write it with your handwriting, you can kind of save time and just write a line separating the two. So it's sort of like almost like a fraction here. So big M with big D on one side, little m with little d on the other side, crossed by, in the test cross, just like we saw a long time ago, test cross, crossed by homozygous recessive for all the genes involved. This individual can make only one kind of gamete. This individual, depending on the nature of the crossing over, gametes are all different sizes. You can have big M with big D or little m with little d. Those are non-recombinant. Or you could have big M with little d or little m with big D, and these are the recombinant. Sorry, REC, recombinant. All right, so it's meant to be a little bit tricky, where you can have to sift through and decide what you're trying to look for. And the thing that is wrong about one of these answers is one of these answers does not have a gamete that could be made by the test cross parent. So in this case, answer D, we know the possible gametes that could come from the heterozygous parent, but those would all have to combine with only one kind of gamete that would be produced by the test cross parent. So this is not possible if you're doing a test cross, because this shows crossing over at the wrong stage. So this individual could have crossing over, but it can only cross over two identical gametes. So two kinds of the same allele. So it's homozygous. If you traded a little d for a little d, you get the same effect. So the only gamete this individual can make is little m, little d. Means that it couldn't produce the offspring genotype on D. Okay. So why don't I stop our video and start a new one here.